Hello, this is Chapter 12, Chromosomal Basis of Inheritance for EFB103. Our topics of emphasis for this chapter are X and Y chromosomes, X and Y linked inheritance, which we'll see is different than can be different than the inheritance we've been talking about, and genetic linkage. All right, so let's first talk about this idea of chromosomal basis of inheritance. And you've probably already got a good sense of this because you know that DNA is where our genes are in our cells, and these are these hereditary factors that Mendel identified. Now, but you have to remember, when Mendel was doing his work, he had no idea that that's what those hereditary factors were. And, even still, he came up with the right patterns and helped explain how heredity works. Those genes that code for all the information that our cells do, those are found at particular locations along the chromosomes. This is kind of a cool image here showing chromosomes at metaphase and using a special staining, basically binding to a very specific gene. And notice you have these glowing spots here and here. There's two, right? Because these are homologs, this guy here and this guy here. And then, of course, these have been copied leading up to the metaphase here in mitosis. And so there's each sister chromatid also has a spot here as well. So genes are found on chromosomes. So research supporting this idea came from a variety of researchers, but here is one example. Walter Sutton here, he was studying cell division, specifically meiosis, and he noticed that during the process of meiosis, you have the separation of chromosomes, sister chromatids, and homologs earlier into different cells and then ultimately into those haploid gametes. And this process of meiosis, as we already know, lines up well with what Mendel described as segregation and independent assortment. So genes are on chromosomes. These factors that Mendel described. This is called the chromosomal theory of inheritance, and it goes further than just genes being on chromosomes. Genes have specific places on chromosomes, and when those chromosomes segregate and assort independently, therefore those alleles, those variants of, say, a particular gene in a particular place on a chromosome, those also independently assort and segregate. And so we can look at the whole process here. This is a figure straight out of your textbook. Have a closer look at that and review it. Make sure you understand what's happening here, where we have multiple chromosomes and these different alleles for different genes, this R and Y gene, and we have a dominant and recessive type annotated here, and we can track their progress through meiosis. Where do they go? What are the possible differences here? We see that independent assortment. You could have the two chromosomes, different chromosomes lining up in different ways. You know about this or should know about this already. And then that segregation right here into the different daughter cells. So let's introduce a new character here in the chromosomal basis of inheritance. This is Thomas Hunt Morgan. He was famous for all kinds of genetic research and really pioneered and expanded using fruit flies for genetic research. How famous was he? Not just for all those things, but he also has a unit of measure named after him. So the distance between genes, one of the units we can use is called centimorgans. That is named after Thomas Hunt Morgan. So what are some of the highlights of the work that he did? And remember, he worked on fruit flies. That's Drosophila melanogaster. Some advantages of using fruit flies as a model organism, kind of like with Mendel's pea plants, you can get a lot of offspring, fast generation time. And where it comes in handy here, where we start investigating chromosomal things, is that they only have 4 pairs of chromosomes. So that's fairly helpful. It's not as complicated as, say, humans or maybe some other organisms where they have more chromosomes to deal with and track and figure out. Another thing as well is Morgan noticed that there were certain types of fruit flies that were just more common. Say, take this red eye example. So red-eyed fruit flies were very common, and they called that the wild type. But there were sometimes traits that were really rare, and these were called the mutant types, let's say. And in this case, we're seeing a white-eyed fruit fly. And so if we do some genetic crosses, kind of like Mendel did, here's what Thomas Hunt Morgan found. So he crosses his red-eyed fruit fly with a white-eyed fruit fly. And what does he find? He gets F1 generation offspring that have the red eyes. So he can conclude that that's a dominant trait. So this seems to follow Mendel's pattern. Nice and easy, right? All right. So here's what happens next. Here's our F1, so all red-eyed flies. We had a white-eyed male in the parental generation. And in the third generation, look at this, a 3-to-1 ratio, right? That's what you'd expect when you do your hybrid cross, monohybrid cross here. You'd expect a 3-to-1 ratio, but here was the wrinkle. Look at this. The white-eyed flies were all male. Now, you might expect if this was random that about half would be male, half would be female, but no, all the white-eyed flies were male. So Morgan had some questions to answer, right? So one option might be, well, maybe the white-eyed females aren't viable for some reason, and that's why you're never getting any white-eyed females in this particular experiment. Well, you can test that. You can do a test cross and you can generate white-eyed females. So that can't be it. And this is really what was happening. It was recognized that fruit fly had this sort of odd pair of chromosomes in males. And this is getting into that X and Y chromosome situation. And Morgan concluded that this pattern that he saw with the eye color gene, it is probably connected to those, and specifically the X chromosome. And so we can now overlay that pattern based on what we know about chromosomes and Mendelian inheritance. So here is our red-eyed female, true breeding. So she has both wild type alleles, so that's indicated by this W+. That plus is usually indicating a wild type allele. And here's that male. The male only gets one X chromosome. It has a Y chromosome too, but that Y chromosome doesn't have this gene. So the male only gets that one allele, and that's going to determine the phenotype. So whatever that one allele says, that's what that male is going to look like. So let's look what happens next. First generation, F1. Well, the females are heterozygous. The males, they can only get the X chromosome from the female parent. If you inherit the X chromosome from the male parent, then you end up with a female fly. So the male offspring get the Y chromosome from the male parent, and therefore they must be red-eyed because they can only get that red-eyed chromosome or allele with that on that chromosome. So now we can see what happens in the F2 generation. We're just going to split our chromosomes into gametes and then bring them back together in our Punnett square here. And you can see that this is the only way to get a white-eyed fly in this particular cross. They are male. So here's this female, red-eyed female, does get that just W allele, but she has the W plus allele too. Therefore, she's going to have red eyes. Now, it's worthwhile noting Nettie Stevens here. She was a graduate student with Thomas Hunt Morgan. He had many graduate students and people that worked with him over the years. But she was the one who had done a lot of the early pioneering work on sex chromosomes. So she studied beetles initially. And noticed that they had these two X chromosomes versus the XY chromosomes. And then so she could apply that to these fruit flies. And then that really helped Morgan then carry out his experiments and make some of those discoveries and bring that stuff together. And Morgan was very complimentary of her. Unfortunately, she died at a relatively young age of 50, but he was always very complimentary of her, citing here one of the most capable and independent researchers in Miss Stevens. So what about those sex chromosomes? Well, fruit flies and beetles have this XY chromosome system. And what about humans? We also have an XY chromosome system. So this occurs, that doesn't mean everything has XY chromosome system. So mammals do, like this, but you look into different organisms and you see different patterns. We'll look at that in a moment. So it's not the only way that biological sex might be determined. And in some ways, it's just happenstance that fruit flies also have an XY chromosome system and help us understand these sex-linked traits. So sex-linked traits, as I just said, those are the traits that are coded for on the sex chromosomes. So the sex chromosomes are the X and Y chromosomes, and the other chromosomes are called autosomes. So everything we've been talking about to this point are autosomes. And interestingly enough, there's almost no overlap in terms of homology between the X and a Y chromosome. There's tiny bits on the end that are similar, but otherwise the Y chromosome is different. But they, again, come as a pair in determining biological sex. So what about those genes? You know, the Y chromosome has the Y-linked genes, and there's not even that many genes on the Y chromosome, about 50. But one that's really important is this SRY gene. That is an important gene that's turned on early in development, and that determines whether an individual develops as a male biologically, or whether they develop as a female if there's the absence of that gene. Interestingly, these two chromosomes can occasionally undergo genetic recombination, like we see with the autosomes during meiosis. So they can line up and swap a piece. And sometimes there are rare cases where an X chromosome actually picks up that SRY gene. So someone could be basically a genetic female having two X chromosomes, but because maybe one of their chromosomes has an SRY gene, might develop male characteristics. All right, so that's just an interesting side note. Genes on the X chromosome are called the X-linked genes, and that is much more like a bona fide chromosome. There's about 1,000 genes, 1,200, I believe, something like that. And a lot of those are just not even related to sex determination. Some are, but some aren't. All right. So in humans, clear sexual differentiation occurs not at fertilization. Well, that is important, but later. It's about the second month of gestation. What's the first event, given your choices here? All right, yeah, back to that SRY gene. That's just critical in sending that development down the path of someone who's phenotypically a male. All right, I alluded to this before, the idea that the XY chromosome system isn't the only thing going in all of the diversity of life, or sexually reproducing diversity of life. So mammals, like us, males are heterogametic. That means males produce two different kinds of gametes. You've got the 22 autosomes plus an X chromosome, or plus a Y chromosome. So that's why males are considered heterogametic. They produce two different hetero kinds of gametes, whereas females produce 22 autosomes plus an X chromosome. Now, there's two X chromosomes, so those are potentially different alleles, but it's 22 plus an X, either one. And so females are homo. Homo means the same, homogametic. All right, so some other things that we see out there. The XO system. This is actually fairly similar to what we see in mammals, and we observe this in some insect groups. The males have an X chromosome, but no Y at all. It's like you just need the one X chromosome, and that says, okay, you're going to be male, and you don't even need that Y chromosome. These guys don't have a Y chromosome at all. And so another option here in birds, the ZW system. So here we see the females as heterogametic, and the males are homogametic. But this idea, again, is the same, that you've got these underlying genetic differences between male and female. And then the last one here is the haplodiploid system. Here, females are diploid and males are haploid. We find this in social insects like this little bee here. And so that's what determines whether an individual is female or male. And if you look back at even the grasshopper here, you can kind of see a similar thing where there's just the one X chromosome, for example. I mean, this is not a haploid individual, but because they have the other autosomes here. But you get a similar sense of this where you just need the one X chromosome, for example, and that produces the male. Okay, so let's talk about these X-linked traits. They follow specific patterns, and because females will have two copies of an allele. They may be the same, they may be different. So here's an example: this carrier mother. So we're just using blue and white here. Carrier, and she can have a son, say here, who's affected because he picks up the X chromosome with the affected allele. She could have a son who is unaffected because he inherits this other X chromosome. So the key here, though, is the male is going to exhibit whatever trait is on that one X chromosome, because that's all they get. And so here's another example: the affected father. Any daughter will definitely get this X chromosome, right? But none of the sons will get that X chromosome, because if the father is going to pass a Y chromosome, that will mean the offspring is a son, and that means the offspring did not get this X chromosome. All right. So some examples of disorders, genetic disorders that follow this X-linked pattern: colorblindness. This is one that's very commonly used as an example. Duchenne muscular dystrophy, hemophilia. So let's look at colorblindness as an example, and let's set this up a bit too. So these are common annotations that we use for sex-linked traits. So here is X to the N, basically, as superscript N. And this is showing you that you're dealing with a sex-linked trait. It's on the X chromosome, and then the allele is shown as a superscript. So we just throw it up here like you see here. Here's X with the little n superscript. In our example here, that's going to be the non-color vision allele. Now, Y is used almost just as a placeholder, an indicator that you've got a male offspring. But notice there's no superscript here. It's because the allele and genes we're interested in are on the X chromosome. They're not on the Y at all. It'd be pointless to put a superscript here because there's none of that gene isn't on there at all. All right, so you can follow the pattern here where we have different parents producing different gametes. We're just segregating gametes and then plugging them back together in a Punnett square. And so if you have an affected male parent, we can see the outcome here, colorblind male parent and homozygous female parent, you are not going to have an affected offspring, but these potential daughters would be carriers. And so you can follow the same patterns here with these different starting scenarios of parents. We can also overlay this onto a pedigree. We've seen these before. We've got an affected individual here, in this case, just affected males. There's no affected females. We don't even need that in the legend here. And notice the skipping the generation. We saw that before when we have a recessive trait. And so that often occurs here as well. And here, like in this example, this daughter was a carrier of this allele. We didn't necessarily know that, but we do once we look at, say, her offspring. So two of the sons here were affected with color blindness, whereas the father of these two sons was not. So she must have been a carrier. So we can track these traits through pedigrees as well. All right, here's a question for you. Red-green color blindness. We know it's sex-linked recessive. We've got our alleles annotated. Two people with normal color vision have a colorblind son. What are the parental genotypes. All right. Yeah, the only way for this to work, if both parents are not colorblind, the father would have to be X big N, so you can cross off C and A. And then the mother would have to have at least one normal allele, so you can cross off B, and that leaves you with D and E. But the problem with D is neither parent here has a colorblindness allele to pass at all. So that leaves you with E. So let's briefly look at Y-linked traits. And remember, there's not as many genes on the Y chromosome, so there are not that many Y-linked traits of interest, but these are going to follow a slightly different pattern, right? So they're only going to be passed from father to son. So daughters aren't going to be carriers at all because they don't receive that Y chromosome. And if we look at inheritance patterns, like we see here in this pedigree, it follows something similar to what we'd expect of a dominant pedigree, right? So because the males are only going to get that one allele, and whatever it happens to be there will be expressed. You don't have another one to sort of cancel it out. All right, so if males only have one Y chromosome, that seems to be enough for an individual to develop and grow and function, depending on your perspective, I suppose. But that's important because if you think of it, well, females have two X chromosomes and therefore it's sort of twice as many genes in terms of dosage. And so there's something called dosage compensation that occurs in female mammals. And a great example of this are these calico cats. So what happens is one of the X chromosomes will become inactivated. And it forms this really dense, unexpressed structure called a bar body. So the chromosome basically like bundles up and none of the genes are turned on. And that means that for those particular genes, females are genetic mosaics. Some cells are going to express one of those chromosomes. So other cells are going to express the other. And so we see that with coat color here is with, say, these ones where the orange X chromosome is inactivated. That's where we see these black patches of fur. And then vice versa, where the orange is active and the black one has formed this bar body. So just another heads up, where we see these white patches in these cats is actually a separate gene associated with that, but it's those orange and black alleles, so to speak, where those are turned on or inactivated early on in development, and it's random which one is turned on or off, and that's why we see these patches. All right, so how does that inactivation happen? So X inactivation is carried out through this XIST gene, X-I-S-T. This is a gene on the X chromosome. It produces an RNA transcript. So we'll get into how genes are expressed later in the course. But the gene is copied, producing this piece of RNA. And this isn't going to be translated into a protein, but the RNA is produced, many, many copies of it, and actually will bind to the X chromosome all along its length. So this XIST RNA will bind to the X chromosome. And what happens, it sort of encourages that X chromosome to wind up really tight and coil. And that's what forms that bar body, that highly condensed X chromosome that is now no longer active. All right, let's switch gears now and talk about genetic linkage. This isn't specifically about sex chromosomes, but really all chromosomes. That's important. And this is another area that Thomas Hunt Morgan was studying. And he noticed that when certain traits he was interested in, he was studying, doing his crosses, he noticed that sometimes certain traits went together. They seemed to be inherited together. And this sort of broke that pattern of that Mendelian inheritance, where Mendel was looking at characters that were not linked. And so Morgan concluded that these were characteristics that were linked, and more specifically, that they were on the same chromosome. So we can look at a map of a chromosome, for example, And here's a zoomed in portion where there are a bunch of genes that are close to each other. And because they're close, this set of genes and things that are close to them, they tend to be inherited together. That's why we say that they're linked. Things that are further apart are less linked. If they're, say, so far apart, they're on a different chromosome, that means they're not linked at all because they're not on the same chromosome. So let's go back to Thomas Hunt Morgan's experiment with his fruit flies. So he was looking at these wild type gray body flies with normal wings. And here's the genotype over here. Again, those wild type alleles annotated with that plus. And here is a double mutant with a black body and vestigial wings. VG for vestigial wings, B for the black body. In the first cross here are the females. We know they would be heterozygous for both of these genes. And then we'll cross the female with this black recessive fly with the vestigial wings. So let's see what this looks like at the chromosomal level. Now remember, we're not talking about sex chromosomes here. So these are autosomes. So each individual has a homologous pair. This is the male here. It is homozygous. It's got the B and VG alleles. And here's the female in that F1 generation who is heterozygous, the wild type and the mutant type alleles. And so here's the offspring you'd expect. So segregation, and then you could put these different chromosomes together and you would end up with these offspring, right? Okay. Here's what Morgan found. So here would be the predicted offspring. Because if you're saying, well, these genes go together, that dependent assortment, here's what we'd expect. This gene's on the same chromosome option here. You'd expect all of your offspring to be either this wild type or a black vestigial, a 1-to-1 ratio. Now, if they're on a different chromosome, that's independent assortment, like we saw with Mendel's studies with garden peas. And you'd expect, in this case, a 1-to-1-to-1 ratio. And so here's what Morgan found. A much different ratio than either one of these. So how does that make sense? What's happening here? These genes seem to be linked, but how do we get these other types that shouldn't occur? If we go back to our chromosomes, you can have B plus with VG plus or B with VG, but there's no option here where you have B plus with VG or B with VG plus. We don't see that in any of the offspring, yet here they are. You have offspring that represent those possible gametes that we didn't expect to see. And so these are called non-parental phenotypes. So these are these unexpected ones. And this is a result of genetic recombination. And so these are also called recombinant types. So offspring with those non-parental characteristics are recombinants. Now, if we see recombinants at a frequency of 50%, that basically means that the genes are on different chromosomes. That's independent assortment. That's what you'd expect just from a study like with the garden peas. If these were unlinked, then you could get gametes with big Y, big R, little y, little r, and then these other combinations too, because they're not linked. They independently assort. And so these are the recombinant offspring here, the ones that don't match the parental phenotypes. So when two genes are on the same chromosome, how do we end up with these recombinant types? Again, it doesn't seem to make sense, but you have to think back to meiosis and what's happening during meiosis in prophase 1, homologous crossing over. So homologs align and they swap pieces of DNA. And this is a way to mix up those alleles. So here's our fruit flies again. Here's our parental generation. Here's our F1 offspring. Everything all looking good, the same as what we've seen so far. Now let's overlay this possible crossing over during meiosis 1. So here are the copied sister chromatids after S phase in meiosis, and then after prophase. Look at this. We've had this one sister chromatid swap a piece with this other one, and now we end up with the B plus VG or B VG plus, which we didn't expect before based on the parental chromosomes. And here, this is why these are called recombinant types, because it takes that recombination event to occur to mix up these different alleles. And so now let's take our recombinant types and our F1 generation, have our cross with our recessive male here, and here is what our possible offspring are. Here's the parental type offspring, and here are the recombinant offspring. And notice that, remember, that ratio didn't come out like we expected because it's these recombinant types, how often they occur is going to depend on how close these genes are together on this particular chromosome. And so we could count them up here, and we get a frequency of 17% of recombinants. And that tells us something about how close these are. And so this allows us to do genetic mapping of where different genes are on chromosomes. Now, the further apart that genes are, the more likely one of those recombination events is going to occur during prophase 1, right? There's just more space in between, so they're more likely to have a break and reconnection in between things that are far apart. If they're really close, the likelihood of a break happening there in that tight spot and recombination occurring is much lower. So you'd actually get fewer recombinants when you were looking at two different traits that are very close to each other. And based on that frequency of recombination, you can get fruit flies of all different kinds and different features that you're interested in, do these crosses and count up the recombinants, and you can start to piece the puzzle together and determine how far apart these different traits are on the same chromosome, or if they're on a completely different chromosome. And with this, we start to build what's called a genetic map. So another thing that's super useful here is a linkage map. And this is ultimately built on these recombination frequencies that we've been talking about measuring. And we can determine a distance in map units. It's important to point out that is not an actual physical distance, but it is an approximation, basically, how far apart these things are based on that recombination frequency. So per 1% difference in recombination frequency gives us one map unit of in our linkage map. Now, if you got sharp eyes, you might notice if we've got three different genes here of interest, B, CN, and VG, that the distance here, the recombination frequency between B and VG is 17%. And then if we looked in these other individual traits, B and CN, we're going to do some crosses with those, and CN and VG, we'll do some crosses with those, and we get 9 and 9.5. Oh, that equals 18%. So how can we have 17% here, but 18.5% when we look at these other pairs of traits together? Well, it comes back to recombination. At some point, usually the threshold is 15% or more. We start to get far enough apart that, yes, there's a greater chance that a recombination event is going to occur between B and VG, for example. But because the distance is great enough, there's also a chance that you're going to have another recombination event in that same prophase 1 that actually takes away the mixing of those different alleles. So it crosses over once and then crosses over again. So the further apart they are, the more likely that is to happen. And so, yeah, 15% is typically the threshold, 15 map units or more. You're trying to get smaller ones. So this 9 and 9.5%, that's going to be more accurate. So you're trying to get things as close as possible to get a better estimate of where these things are relative to one another. That's an important thing. We don't know the exact physical location, but we know relatively where they are compared to one another. So the more traits you examine, the better your map is. What about things that are really far apart? Remember, thinking about that recombination frequency. If things are really far apart, one swap can happen and then another one and another one and another one. let's take opposite ends of a chromosome, for example. Those things are so far apart, usually a big enough chromosome, that they look to have a recombination frequency of close to 50%, which is, again, just like we'd see with an unlinked trait. So traits that are on totally different chromosomes. So things that are really far apart often look like they are assorting independently because they're on different chromosomes. And that's a challenge of genetic mapping, is really identifying what trait is on what chromosome and whether certain traits are actually on the same chromosome if they are far apart. All right, I've got a couple of questions for you. Map units on our linkage map here can't be relied on for an actual physical distance. Why? All right. Yes, it's all about that recombination frequency. So depending on where you're at in the chromosome, that frequency, how many swaps have occurred in crossing over, is going to vary. Let's try another question. So we've got 3 genes now at 3 loci in a species. We don't know where they are in the genome. Each gene has 2 alleles, so sort of this sort of basic Mendelian setup, dominant and recessive situation. We do our test crosses, create our hybrids, F2 generation, and look at the number of recombinants. So, all right, so let's look at gene A, and just one allele, the dominant allele, capital A. It goes with capital B or capital C alleles of these other genes half the time. However, if we just look at B and C alone, those go together 14.4% of the time. So what's happening here? Okay, yeah, so if you've got that 50%, that's a big indicator that you're dealing with unlinked genes if alleles are going together 50% of the time, because that's just half and half, right? So half the time one chromosome's going one way, the other's going the other way. All right, so B and C are different though. This means if big B and big C are going together 14% of the time, that means, say, like little b and big C and vice versa, little c and big B, that means those are going together the rest of the time, suggesting that, we're talking 86, 85.5% of the time, those are linked. So that suggests that these are linked genes. Okay, let's take another shift here to wrap up this chapter, looking at alterations of chromosome number and structure as related to some human genetic disorders. So we can think about large-scale chromosomal alterations. For the most part, these can be very damaging early in development or at fertilization. They're often terminal, fatal, or maybe they can lead to developmental disorders. An interesting note, though, some organisms, like plants, can tolerate some of these interesting genetic changes, and some of these may actually be adaptive in certain cases where we've looked back through evolutionary history of plants. All right, so an abnormal chromosome number, this is called an aneuploidy. Think of, diploid, haploid, that ploid number. We're talking about what's going on with the chromosomes. So aneuploidy is this abnormal number. So how does this arise? It can result from what's called non-disjunction, which sounds like a mouthful, but think of it in a bit of a different way. Disjunction is when those homolog pairs separate in meiosis. So that would be, they become disjunct, they're disconnecting, right? So when non-disjunction is happening, that means that isn't occurring. They're not separating. So as we see here in this figure, you end up with gametes that have basically the wrong numbers of chromosomes. So here are 2 different kinds of non-disjunction events. Here where the homologs don't separate like they should in meiosis 1. So here we have another whole set of homologs in this daughter cell, which you end up with these gametes that have extra chromosomes, or the ones that ended up from this cell, you have one less in each of these. So we have a term for this. a trisomy, so you have like 3 versions of a chromosome, or a monosomy, that would be 1 version of a chromosome, where you should have 2, or 1 particular chromosome. Now remember, these are haploid, of course, they only have 1 copy of each, but these are missing a chromosome. Now you could have a non-disjunction event in meiosis II. Here's where the sister chromatids don't separate, and you see something fairly similar. In this cell here, this was not affected, but in this cell, after meiosis 1, the non-disjunction occurred in meiosis 2, and your gametes you got a trisomy and a monosomy. All right, so some disorders. This is one that probably most folks are familiar with. It's called Down syndrome. This is a trisomy 21. So in this case, a non-disjunction event would occur in either parent, the male or female parent, at chromosome number 21, and the offspring will end up with these 3 chromosomes, and this results in Down syndrome. So this occurs in about 1 in 800 births in the US. The odds actually of Down syndrome increase with age of mother. These are sort of multifactorial things. So, but that is something that it does seem to be a pattern. In terms of a root cause, that's a whole nother story. And so, yeah, this is one of the viable trisomies. Most of the time, if an offspring inherits 3 of one of these chromosomes, it doesn't result in a viable offspring. All right, what about sex chromosomes? So you can have this non-disjunction occurring there as well. So Down syndrome, that was a non-disjunction of one of our autosomes. But here with the sex chromosomes, we see non-disjunction here. And in this example, the female parent. So in this case, you end up with an offspring that has 3 X chromosomes, or an X and no. O means no Y chromosome, or no other X chromosome here. So this gamete didn't have an X chromosome. Again, it has all the other chromosomes, all the autosomes, but just the X chromosome would be missing here. And then you have these other possible combinations. So what does that mean? So there's a few outcomes here. That trisomy X, people who inherit those 3 X chromosomes, no like serious developmental issues. Some studies have shown that females who have 3 X chromosomes tend to be taller, which sort of not really a huge impact. When we get into some of these others, we see more dramatic effects. So an XXY male, so a male with basically 2 X chromosomes, this is called Klinefelter syndrome. You can check all these out. You can Google some of these if you're interested. Monosomy X, that's Turner syndrome. Non-viable zygote would be this OY. So if you don't have an X chromosome, you're pretty much done. You got to have at least that. You can get away with no Y chromosome, but you cannot get away without an X chromosome. These XYY males, so this is something called Jacob syndrome, or which in itself is not clearly well defined as a syndrome. All right, so let's revisit this idea of ploidy. We've talked about haploid and diploid, but you can have organisms that are triploid or tetraploid. And as I mentioned before, this additional ploidy is common in plants, but really not in animals. And this can play a very important role in the evolution of plants. So what are some of the other chromosomal alterations that can occur? Well, let's think about large-scale mutation or damage to chromosomes, and we can lump them into these different categories here. A deletion, and notice our chromosomes just labeled by region here, A to H, just so you can kind of see the parts of it. These are just arbitrary divisions. So here we've lost a chunk. Now, is that important? It could be very important if it contains important genes. If it's a tiny deletion, maybe it doesn't. Maybe that's not a spot with important genes. A duplication event. This, again, well, you haven't lost any genes unless these breaks occur within somewhere important. So this might not have an immediate impact. An inversion is sort of similar where, well, you still have the genes there. They've been reordered. That might be, again, a problem later down the road. Translocation, again, you're still maintaining the same genes. We've got 2 different chromosomes here, but they swap a piece. Now, with these last three, probably these are more of an issue later, or if these occur in germline cells, that, crossing over isn't going to occur correctly later when these kinds of things happen. And so for that immediate individual could be an issue, but probably more important in the long term. All right, so a couple of examples of those. This syndrome called cri du chat is called the cry of the cat, based on the cry that infants will make when they have this condition. And what's happening here is a chunk of one of the number 5 chromosomes is missing. And this is usually fatal in early life. A cancer, chronic myelogenous leukemia, CML, this is caused by a translocation. And so this occurs in an individual. It takes a chunk of chromosome 22, swaps it with a bit of chromosome 9, and you end up with this translocated chromosome 22. It's also called a Philadelphia chromosome, based on where it was discovered. And what's happening here is you look and say, well, you still have all the same genes. What's the problem? Where this swap occurs, it creates an oncogene. Remember when we talked about cancer, oncogenes or proto-oncogenes are really just growth factor genes doing their thing. But when they're turned on full blast, they become oncogenes. They're telling that cell, divide, divide, divide. And that's what happens here with CML. All right, so let's bring together a few things here. Have a look at this question and answer given the options. All right, so yeah, about half the time, the sons would be colorblind and normal height. And we can do this. Remember this trick we learned. If we know the parental genotypes, which we do based on this information here, we can then figure out, well, what's the probability of each of these individual events happening? The probability of an offspring having little d, that's 50%. The probability of them having x, little n, y, that is 100%, because the female parent is this genotype. And so it's just a half times 1, which is a half, 50%. All right, so that's it for this chapter. We'll see you next time.